So my name is Alison Düttmann. I'm the incoming president of Foresight Institute, and we are a nonprofit to accelerate technologies for the long-term benefit of life. We usually do that via three types of meeting. Some are technical competitions where we actually try to push technologies that have a potential long-term benefit and try to use foundational science and tech to solve a specific problem. Healing the planet and healing human bodies. So health extension are usually like our main go-tos here. And then we have a second type of meeting that's private strategy meeting. So this is mostly thinking about policy related to technology. For example, on AI and artificial general intelligence, we have an annual meeting there. And then the third type of meeting that we usually do is the public salons. And so those are educational. We like to think in our community. And those have been going on usually once a month. We had just launched a longevity bi-monthly event at the beginning of this year, and then COVID hit. And so since COVID hit, we've been really ramping up our third type of event, which is the public event, and that's mostly largely because we're trying to we're trying to help people a little bit better to make sense what's happening with COVID-19, to showcase projects that are currently ramping up their support, and this is definitely one of them that we're going to talk about tonight a little bit. And then the third one is we're trying to use this as an opportunity to already think about long-term change that we could be pushing for leveraging the current crisis. So currently it seems like we're doing a lot of all three of them, and starting next Saturday we're going to launch into the next phase of this experiment. We're going to still be continuing to do daily meetings, but starting next Saturday, they're all going to be a little bit more structured. So it's more going to be a workshop where we're trying to really tackle one specific aspect, which popped out of those salons, and try to see whether we can actually get a working group together to think more clearly about what are actually potential action items that we could be launching into right now, and to potentially hold doors open that have been opened with COVID-19, and to try to create some more long-term change. I'm going to share the schedule in the chat right now, because you can already look on the schedule that you see a little bit about the programming that we have going on starting next Saturday. So this would be a little bit more hands-on, I think. And so feel free to check that out. Meanwhile, we're still going to continue the normal salons next week before we're going to start the actual working groups. Okay, cool. I'm going to share that in the chat right now. And there's a lot more people on the waiting list. Man, this is a very high demand salon on a Saturday. This is great. Thank you so much, John, for being so flexible and joining. Your project was sent to me now from four different corners, as one highlighted, of a lot of people thinking that if there's one project that has a really good angle right now at actually doing something useful on a reasonable timescale, then it's yours. So I'm really happy that you were able to so spontaneously join us today. And I won't be taking away much more of your time. I'm terribly excited for the presentation that you're going to give. So I encourage everyone to maybe wait until the presentation is over. The slide, we collect questions in the chat, and then people can launch into a discussion, and I'll cue the questions. The topic of today will be accelerating development of pan-corona vaccine and therapeutics using breakthrough genetic medicine. And I'm going to share some of your bio in here. It's quite extensive, and yeah, I can't wait for what you have to share with us. And yeah, with that being said, I'll leave the stage up to you, and I'm going to be taking questions afterwards. So feel free to immediately start collecting a few questions, and we'll queue them up. Thank you so much for joining.
Okay, Alison. Yeah, thanks so much for inviting me. It was it's really my pleasure today to join everybody. I'm with everything that's been going on, and our lives have been completely transformed. it's a scary thing that the world is going through. We at Aegis Biodefense feel very fortunate that we have a technology we think can address it in short term. I'm excited today to share with you what we've done so far and what our plans are for the near future. So Aegis Biodefense is a brand new company. I'm also involved I'm a professor in a Canadian university. I've spent my life developing nanoparticle technologies and genetic delivery technologies. And so we were perfectly poised with the development of a key technology that will enable the rapid prototyping of a vaccine candidate for COVID-19. And so Aegis was just born out of this need in the last 2 months. But as you'll see, there's much more to it than just COVID-19. Okay, so I don't have to tell anybody in this group that we're facing this unprecedented threat. Although many people, predicted it, it's sort of taken the world by storm, and we're finding that it's having impacts that are going to be very difficult to come back from. Not only the economic impacts, but the impacts on humanity. And its global spread and economic impact are completely unprecedented. So most of you, have been watching the news and reading. It's caused by this infectious coronavirus, this novel SARS-CoV-2 coronavirus. And of course, the number of infections and deaths are rising exponentially in Canada, US, and worldwide. And the impact to our health system is unprecedented and growing with no end in sight. So really what we need, we're thinking about repurposing drugs to be able to ease the symptoms. Several of them have been proposed, are currently being tested, and we really don't have a lot of evidence that any of them are working particularly well. Vaccines are really not positioned to be able to, traditional vaccines aren't positioned to be able to address this. 5 to 7 years average approval rate for vaccines, and to make a traditional vaccine against SARS completely failed. So we're in a situation where we need some new ideas. And so really we're faced with this problem that we need to get a solution out to people quickly. So every day that we wait, is thousands of deaths, and as it spreads exponentially, and we're taking these unprecedented measures just to prevent the transmission. So unfortunately, current vaccine technology can't really respond to these kinds of emerging, rapidly spreading threats. The traditional vaccine approach is either called a subunit or a virus-like particle vaccine. This is where we either inactivate we get some of the COVID-19 virus and we inactivate it, maybe using formaldehyde or UV light. And then we inject a tiny bit of it into the patient, and hopefully the patient will produce an immune response that'll protect them from infection from the real virus. Unfortunately, you have to get the manufacturing figured out. You need to do testing, first in animals and humans, and typically these take about 5 to 7 years to develop and validate. So even in a situation now where we're exceptionally motivated, we're dumping tons of money into manufacturing, we're still looking at 9 to 10 months before the first of these vaccines that have had any thought put into them are even available for animal testing. And so we're in this age of technology that's rapidly moving forward. So tools that we have at our disposal that we didn't have before are genetic or genomic medicines. Now that we've sequenced the human genome, we've sequenced the coronavirus genome, very early on in its spread, we have the ability now to figure out what makes the coronavirus so deadly, and also rapidly come up with genetic means to be able to fight the virus. And you've probably heard a lot in the news about RNA vaccines. Companies like Moderna and CureVac and BioNTech have created RNA vaccines that basically deliver the instructions to make the coronavirus spike protein into the human cells. The human cells make that spike protein, and then hopefully the body produces an immune response against it. Unfortunately, RNA vaccines, while they have all the benefits of rapid prototyping and rapid transition into clinical trials, RNA itself is extremely expensive to manufacture. RNA is in the body is designed to be transient, so it's very unstable. And even with the best chemistry to stabilize it, it's quite a bit less stable than other solutions like DNA. And unfortunately, the solutions, the delivery technology that the companies are using, like Moderna, are actually relatively toxic. And so their maximum tolerated dose, even for a vaccine intramuscular injection, is quite low. So their window of opportunity to basically make enough protein to generate immune response is very, very narrow. On the other hand, DNA vaccines have had, there have been a lot of companies working in the DNA vaccine space, and really DNA vaccines have suffered from a lack of effective delivery. And so the most popular, the most efficacious DNA vaccine is being put forward by a company called Inovio. Inovio has a DNA vaccine that they've shown to be effective in animals. Unfortunately, to introduce the DNA into the patient, they require a handheld electric device that delivers an electric shock. So we're going to have to manufacture and distribute these electric devices to every single site where we're going to give this vaccine. So even though we know a DNA vaccine can work, it's just really not feasible for the scale that we need right now. So Aegis is coming to the market with a solution that basically combines the best attributes of a genetic medicine with the stability, ease, and cost of manufacturing of DNA without the electricity, right? So we have a proteolipid vehicle delivery platform that is best in class for delivering DNA into patient cells so patients can make their own medicines. And so it's called Fusogenix. You may have heard of it. We're partnered with a number of other companies. So we developed this technology in my laboratory in Canada. We created a company out of the university called Entos Pharmaceuticals, and Entos has been working with longevity companies, with oncology companies, with about 18 companies in total developing sort of the next generation of genetic medicines. You may have heard of Ocean Biotechnologies and Oncocenix, and now working with a select group of founders that come from these groups. We've started Aegis Biodefense to develop solutions for COVID-19, but also solutions for infectious disease in general using this rapid response platform. And so in addition to having all the benefits that I mentioned of ease of scalability and inexpensive and rapidly prototypable, we also have the flexibility within DNA to have a cargo or payload size that's large enough to provide pan-disease protection. And this is really important because Bill Gates and others have predicted that we're going to suffer from a pandemic outbreak of coronavirus a while ago. So we have the opportunity to create a vaccine that'll work not only for COVID-19, but also for any other coronavirus threats by generating immunity to the key structural conserved proteins in the coronavirus. And this is really important because this is a key differentiator for our technology. So even if we're not the first, say Moderna's a little bit ahead, even if their vaccine works a little bit and they start to distribute it, we can come to the market very quickly afterwards with a solution that'll be much more compelling for people to purchase and give to their populations because it will protect against future threats as well. And so we've already spent a lot of time. We're already 2 months into this. We have this Fusogenix technology to deliver DNA. Because of DNA manufacturing and microfluidic manufacturing for our platform that's already ready to go into cancer and a number of other indications, we're ready to scale this up to manufacture hundreds of millions of doses. We've got some great animal data. Our platform produces high expression of these proteins, plus has excellent safety and tolerability. And we're ready and we've actually planned and we're ready to start a phase 1 clinical trial in less than 2 months. And so, there are many other players in the COVID-19 vaccine and therapeutics space. And so we're going to win because we're going to be the first to market with a pan-coronavirus DNA vaccine. This is really key, and I explain this. We're really the only company that possesses both this superior payload design flexibility and the fact that we can rapidly prototype DNA, and of course the delivery platform that allows us to create a formulation that we can inject in the arm and intramuscularly, just like the flu vaccine. And so because this technology has been used for a lot of other applications, it's a highly de-risked platform. It'll enable us to advance multiple programs in parallel to maximize favorable clinical outcome, not only for COVID-19. So we have a couple dozen COVID-19 candidates that we're already testing in animals. And then of course, because of the nature of this current pandemic and FDA and Health Canada and other regulatory agencies really want to accelerate the delivery of solutions to the public, we're going to be able to start our first human phase 1 in less than 3 months. We should be able to complete our phase 1 trial in 6 months and actually apply and start our phase 2 trial before the end of that phase 1 trial. And if everything goes right, and I know this is a bold statement, I think we can get to market within a year. So again, I explained a little bit about why DNA vaccines I think are very compelling. So I mentioned DNA actually delivers the instructions to make part of the coronavirus in the patient's own cells. And so because DNA is easy to make and inexpensive to manufacture, we can design, test, and manufacture a clinical scale in just a matter of weeks. And it's really, I did a little bit of comparison to other people in the space, but really people have been trying to develop DNA vaccines for years. And it's been very difficult to deliver the DNA inside cells. And the current platforms that companies are using to deliver messenger RNA do extremely poor job of delivering DNA. And so that's how we're different with our platform. And like I said, Inovio uses electric shock. CanSino actually took a different virus and put the COVID-19 instructions inside a different virus. So this may work, but then manufacturing and scale up is extremely expensive. And it may work for parts of China, but it's going to be very difficult to translate that over to North America. And so our Fusogenix platform, like I said, we can deliver intramuscularly just like the flu shot. And we already have a GMP clinical grade manufacturing facility up and running in Edmonton, Canada. And so I mentioned, comparing to some of the competitors and to repurposed drugs. I won't go through this anymore, but I want to highlight a couple things on this slide. Again, the ease of large scale manufacturing and the lower cost of manufacturing, the stability I mentioned. One thing I didn't mention is because with a messenger RNA payload like Moderna and CureVac or others are using, they have a limited amount of instructions they can put into their vaccine. So because we're using DNA with unlimited payload size, we can be very creative in what we put in. So we can think about things like expressing neutralizing antibodies or putting neutralizing antibodies that have already been discovered to cure COVID-19 in our platform. We can do competitive inhibition. So we can make small peptides that interfere with COVID-19's ability to bind the receptors they use to get into cells, the ACE2 receptors. And we can express epitopes or immunostimulatory parts of the proteins from multiple different proteins in the coronavirus to really enhance our chances of getting that robust immune response that can carry over to other coronaviruses. Because we're not using a live virus, we have no risk of infection. Again, we can protect against future coronavirus. And the other thing that people are talking about, you may or may not be familiar, is what's called an antibody-dependent enhancement response. So when vaccine companies were working on the SARS vaccine, one of the reasons why SARS vaccines failed is because when they used the full spike protein in their formulation, they actually produced antibodies in primates that helped the virus get in cells. In fact, there were a couple primate studies where they were testing out these experimental vaccines where all the primates died when they would challenge them with SARS. And the reason was this antibody-dependent enhancement response. So because we're able to rapidly prototype dozens of candidate vaccines at the same time and engineer them in different creative ways, we'll be able to rapidly test and come up with one that doesn't produce this antibody-dependent enhancement response. We know companies that were first to the poll, like Moderna, didn't have time even to do any animal studies to validate this. So that's definitely a risk for them. Okay, so what makes our technology platform different from a CureVac or a Moderna's platform? Well, we already know that viruses do a great job of getting inside cells and putting in their RNA or DNA. Viruses like herpes and HIV and others have developed and evolved these fantastic fusion proteins that do a very good job of going through the body, finding a cell, say in the lung, engaging with its membrane, and then mixing the lipids from the virus and the patient cell together to inject their DNA payload. And so if you look at sort of all the viral fusion proteins from all these different viruses, unfortunately, they're not really manufacturable. They're very large. They have these huge, what's called ectodomains, these domains on the outside that in and of themselves are immunogenic. So the body will recognize these and get rid of them. And unfortunately, you won't get your vaccine to actually work. And they're impossible to manufacture. So fortunately, I had the fortune of meeting a researcher, a virologist, a Canadian virologist named Roy Duncan about 15 years ago. And we've been working together since then. He discovered this class of orthoreoviruses that actually infect alligators and birds. And what's really interesting about them is they don't use a fusion protein to enter the cells. But once they get inside cells, they cause all the cells around them to rapidly fuse together. And so he was like, oh my gosh, Nobel Prize moment. He went and said, okay, I'm going to figure out what this new fusion protein is that nobody else has discovered. It actually took him 3 years because it was so tiny. It was being excluded from all the ways he was trying to look at it. So when he finally figured out this is a really tiny bugger, then he went back and discovered now what's known as the smallest known fusogenic protein by 2 orders of magnitude. So this is really, really tiny. And believe it or not, it's sufficient to fuse 2 membranes together, or importantly in our case, to fuse a DNA formulated into a vaccine with a patient's cells to express the epitopes. And so he went on to look at all these different fusogenic orthoreoviruses, and I've been working with him over the last 8 to 10 years to take the best parts of those fusogenic proteins and create a highly engineered proprietary fusion protein that forms the basis of our DNA delivery technology. And so the Fusogenix DNA vaccine platform is basically just a combination. It's a proteolipid vehicle. So it's composed of lipids similar to other platforms, nanoparticle platforms. Instead of using, so other lipid nanoparticle or nanoparticle platforms in their lipid composition, they use a positively charged lipid to disrupt the membrane of the recipient cell and basically punch a hole to get in. And so positive charges actually work pretty well in disrupting membranes. And so nanoparticles formulated with these positively charged lipids do a pretty good job of getting DNA and RNA in. The problem is that they also, poking holes in cells is quite toxic. So at a very, very low concentration, you get these dose-limiting toxicities with traditional positively charged lipids that make it very difficult to dose at the levels required to express enough protein to get an immune response, or for many other therapeutic indications. And so by incorporating these fusion-associated small transmembrane proteins into our Fusogenix platform, we can take out the positively charged lipids, we formulate it with naturally occurring lipids that are already in your body, fats that are already in your body, and then the fusion protein does the delivery part extremely well. So we've optimized formulations just for DNA vaccines that have high expression of their payload. So in this case, expressing a wide variety of different spike and capsid proteins. They're significantly more efficient than a lot of the viruses or non-viral formulations that are out there. And because we're just making it with lipids and protein and DNA, we can make it using a scalable microfluidic manufacturing platform that we can scale up, all the way from the benchtop to hundreds of millions of doses. And I'll get into a little bit more into that. As far as our design on the vaccines, we've been working, since basically the beginning of the year. We knew this was going to be a pandemic. Initially looking at the full spike protein and the full nucleic-acid protein, the N protein, and looking at the ability of that in our platform to produce an immune response. Then we played around with optimizing the spike protein, just looking at the interface between the spike and ACE2 receptor, working with some key partners, including a company called Epivax, that's a Rhode Island company. They are well known in the immunogenicity business for developing really, they have in silico algorithms for figuring out not only what are going to be the most immunostimulatory epitopes, but most importantly, can remove the regulatory epitopes that will dampen the immune response. And so we've been working with them to generate a number of different things. So spike proteins that have a decreased chance for antibody-dependent enhancement, decreased chance of immunoregulatory epitopes, and optimized epitopes that will produce the robust immune response. And we've also been working with them to create what we call a string of beads. This is basically using in silico techniques that they've developed to identify epitopes from all of the different coronavirus proteins. So actually four of the most important ones. So E, M, N, and S. And then so that will give us not only the ability to generate what are called neutralizing antibodies to block viral uptake, but also to produce a cell-based response. So a T cell killer response that will should have a dramatic effect in active infections to reduce the disease severity. And so we think this is really important to create both an active protective and therapeutic vaccine. We've already started animal testing. So we have a large group of collaborators and partner companies that we're working to do testing. So obviously the US and Canadian governments have funded a lot of this work already. We're in mouse studies already looking at immunogenicity. We have plans to go into ferrets, which is a validated challenge model, so we can show that we can protect ferrets from getting COVID-19. And then also we're planning in an IND-enabling study to do a study in primates as well. And this is all teed up and ready to go as soon as we've selected our lead candidate. So as I mentioned, I mean, this is Aegis Biodefense is going to be working intensely on COVID-19 in the short term, but this is really an infectious disease play. We're going to use this rapid prototyping technology to go after a number of high-value targets, developing assets, again, first for pan-coronavirus, and using basically the situation to rapidly scale and get to market our first product. And then working on other neutralizing antibody approaches to coronavirus and things like HIV, working on a pan-influenza vaccine and therapeutic using the same approach, and pushing these forward. Like I said, we've already established the key partnerships we need to be able to achieve the aggressive timelines we looked at. So we're working with another Canadian company, called Precision Nanosystems. They've developed this microfluidic technology. They're working with us building the GMP commercial manufacturing system. We're working with Epivax, as I mentioned, in Rhode Island. We're screening vaccine candidates. So as you probably know, labs around the world are all shut down. Basically, research that is not involved in COVID-19 has been completely shut down. We have a fantastic 20,000 square foot lab, fully open, completely devoted to COVID-19 work. We're recruiting patients in an ethics-approved clinical trial to collect patients both who are naive, who are undergoing current COVID-19 infection of various degrees, and convalescent patients who've already recovered from COVID-19. And part of our screening process will be to screen our candidates using their blood to look to see if patients who've successfully mounted immune response against COVID-19 are producing that immune response against the epitopes we've chosen for our vaccine. We're already testing in animal models in Dalhousie University, and we'll be working with one of the only Canadian BSL-3, so level 3 facility that can use COVID-19 and has the challenge models in ferrets. And again, we're working with a company called Versio. They've got the animals all ready to go as far as our non-human primate challenge study. And this will be key both for FDA and Health Canada. If we see any antibody-dependent enhancement response in, say, Moderna's clinical trials or some of the other clinical trials that will be starting soon, this will be key to have this data to demonstrate that we won't get that. And again, we've already designed and are ready to launch phase 1 and phase 2 COVID-19 DNA vaccine clinical trials. And sort of this is the timeline we've mapped out, and so far extremely achievable. I mean, it's dependent upon sufficient protection in our animal studies, and then the plan to start phase 1 clinical trial in about 2.5 months, building up our GMP manufacturing facility so that we can deliver doses for phase 2 and then commercial manufacturing. I'm going to spend a little bit of time talking about the platform. So I've told you that it's better, but I want to show a little bit of data that supports that. So one thing I mentioned is the tolerability toxicity. Thinking about positively charged nanoparticles compared to the Fusogenix proteolipid vehicle. So this is just an in vitro sort of putting cells in a dish and asking the question, as we increase the concentration of a DNA payload formulation, what do the cells do? And you can see, the red line is a positively charged nanoparticle. It's fine at low doses, and we know that these formulations are fine at low doses. As soon as you increase the dose of a DNA payload, it's extremely toxic. All the cells die. So with our formulation, it's basically completely flat. We don't get any cell death, even at extremely high doses. If you look on the right side on the expression, at low doses, again, our expression depends on the cells. Certainly for some hard to transduce cells, we're quite a bit better. For the general cell lines we use in the lab, about the same. But as we increase that dose, basically all the other cells are dying, and we're maintaining a high level of expression, which is important because a vaccine is a locally administered therapy. We're getting a very, very large dose in a very, very small area. So toxicity is important. And so this is a direct shot over the bow of our competitors. So this is comparing the tolerability of the Fusogenix platform compared to what's called MC3 or SNALP. It's got a bunch of different names. That basically is the platform that our competitors like CureVac, BioNTech, and Moderna are using. So they're using mRNA formulations, which we know it's published clinical data that they have very, very low maximum tolerated doses, even in an intramuscular injection. In this case, we're delivering a DNA payload systemically, which is quite a bit more toxic because the liver concentrates it typically. Even at a 50 microgram dose with the competing formulation, none of the mice lived more than 24 hours. In fact, we put the dose down to 2 micrograms, of just a fraction of that, and they all died. So we had to go down to a sub 1 microgram systemic dose, so they didn't die. Our platform filled with DNA, 600 micrograms, so 600 times at least less toxic. We're getting great expression throughout the animal, and these animals are just fine. If you look at we got pictures of their livers down there. You can see that these MC3, positively charged lipids just do a number on the liver. So the liver's got a whole bunch of these microemboli, and it's one of the main reasons for death in these animals. As well as, I'm showing you this bar graph here. All I want to say is that there's 3 lines on each of those bar graphs. Each one of those bar graphs represents a cytokine. You've probably heard of cytokines. The cytokine storm is what seems to kill patients that have COVID-19. It's basically your immune system getting over-revved and releasing all these signaling molecules, and it can it can be a deadly situation. And so on the left side there is just delivering a saline solution. We don't get any spiking up of cytokines. In the middle, we have the competitor formulation that you get these massive spikes in cytokines. And then our formulation on the right, we get virtually no spiking of the key cytokines that cause toxicity: IL-6, TNF-alpha, interferon gamma.
Wait, could you repeat that?
Which part?
The last part about the inflammatory markers.
Yeah, sure. So in this case, this is the same mouse experiment. So we dosed mice systemically, in this case with a 150 microgram dose, and looked in their blood at the levels of cytokines, and looked at this panel of cytokines. So interleukin 1 beta, interleukin 4, interleukin 5, interleukin 6, interleukin 10, CXCL1, TNF alpha, and interferon gamma. And we see several logs of spike with the competitor's formulation that we did not see with our formulation.
Thank you.
We've done, because we're working with other programs with other companies as well, we've got a lot of data around the biodistribution of these formulations, the safety of these formulations, where we've given in primates, for instance, about 20,000 times the dose we'll need to give for a vaccine, and seen no adverse pathology in the major uptake organs. So we know that it's, certainly in the concentration we're going to use for a vaccine, it's going to be extremely well tolerated. We've done a lot of work around producing anti-drug antibodies in animals. So even though we're using a viral fusion protein, we initially expected that it would elicit these anti-drug antibodies. We don't see, even at very high doses, we don't see that. We've also looked at other immune-related assays to look to see if we're getting a non-specific immune response against our delivery platform. We don't. And again, we've done a lot of IND-enabling studies in primates, in canines, in pigs, in mice and rats, and we don't see any adverse pathology or anti-drug antibodies or histological changes, even extremely high doses. I like to show this slide because it really hits home the challenge that the industry has had with these positively charged lipid particles. And these are all companies you may have heard of. Most of them have gone out of business because these are published clinical data for different trials. These are systemic dosing, not local dosing. You can see that the positively charged particles are extremely toxic. These 3 in the middle, so Marina Biotech and Arbutus and Alnylam. Alnylam actually has an approved product, a SNALP product. Its maximum tolerated dose is 0.9 mg per kg, which is still extremely low. We already know, we've tested ours systemically. We couldn't find a maximum tolerated dose for our formulation, so we know we're extremely well tolerated. And so when we make a plasmid DNA formulation, we can deliver it through multiple routes. So I'm showing here, this is plasmid DNA encoding just a gene that gives off light, luciferase. And so we inject it in the bloodstream, we see luciferase, basically the whole body lights up. We inject it into the muscle, we see very, very strong expression in the muscle, even at low doses. And interestingly, even when we give it orally through orally in the mice, we see really strong expression. So we're going to also look at the ability of the vaccines to be delivered orally, which I think will be a key differentiator as well. Repeat dosing can be extremely important. So I mentioned these anti-drug antibodies. So for any VLP or viral-based approach, you're going to get a really strong induction of anti-drug antibodies. So you're going to have to give, ideally the ideal dose first. We've done, many multi-dosing studies. In this case, we're giving intramuscularly every month for 5 months, and we don't see any reduction in expression of the antigen after 5 months. In fact, we see the antigen expression just gets stronger over time. Manufacturing is going to be key. So let's say we're 2 months down the road, we've got a fantastic vaccine that produces the right neutralizing and cell-based responses. We've got to rapidly scale up our manufacturing, not only for clinical trials but for commercial deployment. And so, like I said, I mentioned we're partnered with Precision Nanosystems. We've got we're fortunate that we have exclusive access to I would say we've had a few consultants come in. It's the very best GMP manufacturing facility in the world that I've seen anyway, and that many of our consultants have seen. We have access to 5 complete clean rooms, so we can manufacture hundreds of millions of doses out of those rooms, and we're working with Precision Nanosystems. We've already got the facility built. We're just qualifying it to produce the clinical doses, and then building out the capacity shortly to have first commercial product. I just want to spend a little bit of time. Many of you may or may not be familiar with another company, Fusogenix, that is using the same Fusogenix platform in their DNA-based suicide gene therapy platform for cancer. Maybe I didn't mention, but in I'm also a professor of oncology. I'm really, really passionate about potential cures for cancer, and so we've been driving forward this Fusogenix platform. And this is entering human clinical trials at the beginning of next year. So I just want to spend a little bit of time showing you, how well this platform works for delivering DNA. So in this case, this program involves basically killing cancer cells based on what they're thinking. And so it seems like a crazy thing, but a cancer cell knows it's cancer because it has oncogenes that are activated that aren't activated in any other cells of the body. And using a plasmid, we can use an engineered promoter to basically hijack these signals that are basically telling the cell to grow, and instead short circuit that and tell the cell to kill itself. And so we basically have an engineered promoter that's receptive to certain oncogenes, and then we redirect that signal to express a caspase-based death protein that rapidly kills the cell within about 4 hours. And because now we're specifically inducing the production of these suicide genes in cancers, we can also piggyback on additional proteins to clean up the rest of the tumor. So in this case, immunostimulatory proteins like CD40 ligand. And so we've got a number of assets that we're moving forward to clinic based on this idea of delivering a systemic gene therapy that won't have any effect in any cells except for the tumor, and will specifically tell those cells to kill themselves. And so I'm just going to show a couple slides on that, just to give you, sort of an idea of the power of this delivery system. So on the left side here, we've got an animal study where we're giving on the left side, just a ubiquitous DNA plasmid. So in this case, it's driven by a strong promoter that expresses everywhere. So you can see when we give it a systemic injection, it's going everywhere. It's in the brain, it's in the spleen, it's lots in the liver, lots in the lungs. But there's no cancer cells anywhere but on the side of the animal. So then when we go back and we give a formulation that's only driven by this oncogene promoter, we can see that we already know a ton of it's going to the liver, lung, and everywhere else. But we see they're completely blank. We don't get any expression anywhere but in the tumor. And in fact, this is a big experiment for us because the expression of the tumor is so high that we can see the light from outside of the animal. So it has to be really high for that to happen. And so this is why we're getting this highly selective killing of only the cancer. And so it works really well. We've looked, we're going to phase 1 with prostate, colorectal, lung, and pancreatic cancer. This is just an example of data from prostate cancer. We're able to, with a single injection, basically remove over 90% of the tumor volume in just 48 hours. And then with multiple injections, completely cure the animal. In this case, so we'll be going to clinic again next year. Many of these, I'm sure many of you heard of checkpoint therapy or checkpoint inhibition. So now these are drugs that take the brakes off the immune system to recognize cancer. This has been transformative for many patients. So melanoma, for instance, up to 38% cure rate, durable cure rates. The issue is that 30% is great, but what about the other 62%? And many cancers like prostate cancer have had very little response to these checkpoint inhibitors. So we think this would be an excellent, completely coming up from a completely different angle to both work as a single agent and in combination with checkpoint inhibitors. So in this case, we're looking at an animal with an intact immune system with a colon cancer. We're giving either a single agent with this DNA-based gene therapy. We're better than checkpoint inhibitor with a single agent. In combination, we cured every single animal. So we've done, and the animals that were cured, interestingly enough, when we re-challenged them with a tumor, they had a durable immune response to that agent, or to those cancer cells, and did not regrow tumors. So this is going forward to the clinic next year. Initially, this is going to be the first in human Fusogenix clinical trial. And now Aegis Biodefense is going to take this platform to clinic before that. So we're very excited about that. We've got a great team. So as I mentioned, I've been a translational oncology, but nanotechnology researcher for many years. I've built a large translational research program where we do all the way from discovery to clinical trials. Working with Han Liang, a fantastic entrepreneur in her own right, with several really successful companies and exits, and also founding with Roy Duncan, the discoverer and inventor of the Fusogenix technology. We already have a big lab in Canada, 20,000 square feet. We actually have 2 locations there. We're in J Labs in San Diego, and all these labs are open and ready for business. And we have a great team of scientists already working on this. And just to give you an idea, we're building, we're in the process of building out just a spectacular team. Got regulatory and quality already, a CSO, business development, preclinical development, and a variety of really dedicated scientists that are maintaining the adequate space in the lab, but are really passionate about making an impact on this COVID-19. And so we'll finish off there. Fusogenix is really the key to this. It's a highly differentiated vaccine and therapeutic technology. We're going to bring forward, we have these 24 candidates that are both vaccines and active therapeutics against COVID-19, and we're looking forward to scaling that and getting out of the market quickly. Currently, one of the reasons why we rapidly want to get this out there to you, we're raising a seed round of financing. Please email me or call me if you're interested in contributing to that. With your help, I think we can bring a DNA vaccine to the population within a year. And with that, I'm happy to take questions and comments.
Thank you so much. Man, you covered a lot. Awesome. I would love, if you're willing, if you could share the slide decks or at least your contact info, then I can put it in the schedule afterwards for people to look up. I think we had a few questions collected already. I also have a few, but let's hear from Creon first and then Alyssa.
Oh, Creon, you're on mute.
Sorry about that. Yeah, just a sec. Okay, I have a couple of questions. One, sure. Does your talking specifically about the COVID-19 vaccine candidate, does it need an adjuvant? Have you worked on that? Where are you at on that?
Yeah, so typically with a DNA vaccine, you won't need an adjuvant. We have, we have several sort of de-risking backup plans. One of those backup plans, we're partnered with Michael Houghton, who developed hepatitis C vaccine. So he has a subunit vaccine for COVID-19. So in the case that we do, need an adjuvanted subunit therapy, we'll provide a boost to the initial DNA vaccine prime a couple months after with the protein. So that's sort of the worst case scenario. But the best case scenario is that single injection DNA vaccine produces the required immune response. And that, based on the data we have so far, it looks like that'll be the case.
Amazing. Okay. And then second quick one. I'm not so much questioning your approach here. I'm asking to be educated about the comparison to another approach, which is, why is no one seen to be touting or pursuing an attenuated virus vaccine? Everyone wants to do recombinant. No one wants to do it the old-fashioned way. Is there a good reason for that? And if so, what are they?
Yeah. So people are definitely working on attenuated virus vaccines, although they may not have gotten a lot of press. Certainly that is a tried and true approach that we know has a great chance of working. the real challenge with that is speed. We know everybody's out there saying we might have a vaccine within 12 to 18 months. An attenuated vaccine can't be manufactured and validated and gone through trials with that speed. And like I said before, the typical approval rate for those kind of vaccines are 5 to 7 years. Influenza, flu is a little bit of a different situation because they have everything all set up and ready to go every year. And they make very small changes to their vaccine, and they make some guesses as well, which are sometimes wrong. But I think that's more the issue. Yeah.
I thought flu is recombinant now anyway, because they're trying to do it against multiple strains. Or is that?
So it'll be a recombinant VLP. It's an in-between approach. So it uses a virus-like particle.
I see. Thank you. I have another question or two, but I'll wait.
Sure.
All right, Alyssa, do you want to chime in?
Yeah, can you hear me?
I can.
Cool. So first, thanks for your talk. It's excellent to learn about such an exciting project. And, I'm sure everyone is wishing you the best of luck. Everyone wants to get rid of the pandemic as soon as possible.
Yeah,
so everyone in the media has been saying, oh, it'll be 12 months to a vaccine, 18 months to a vaccine, 12 months to a vaccine. It sort of just gets repeated over and over. But I haven't seen a good breakdown of, here's why it's going to take that long. Why not 3 months? Why not 6 months? Why not 10 years? And so on. Do you know of like a good analysis of, where that timeline comes from exactly?
Absolutely. Yeah. I mean, it's the clinical regulatory pathway that's laid out in most industrialized countries that dictates that timeline. Some of it is de-risking. So obviously you want to proceed forward with a candidate with the least amount of risk because these clinical trials are extremely expensive. But a part of it is just an abundance of safety and caution. So I mean, we've already seen some really innovative ways of starting trials. So like I said, Moderna has started an RNA vaccine trial already. CanSino in China has started a vaccine trial. They've really cut a lot of corners. In fact, they started their phase 2 after only 7 days in a phase 1. But typically it's more the follow-up. So typically with the phase 1, you say you do 2 doses, one at day 1 and one at day 28. Typically you have to follow those patients for 6 months to make sure nothing happens after 6 months. So a phase 1, by the time you get your readout and submit your data to the FDA, could be a year and a half before you're done. And that's just the phase 1. There are 3 phases of development, and each phase gets more complicated. Phase 2 might be hundreds of patients, say 300 or 400 patients. Again, to recruit those patients, do all the analysis and everything, make sure it's correct, get the manufacturing. Again, that could be another 2 years for your phase 2. Phase 3 is even more complicated. You have to show it in a general population that you're as good or better than any other options. I guess with COVID-19, there are no other options. But again, it's just it just takes time to get it all done, and then you have to look at all that data and decide whether you can go, be approved to go commercial. So I think, in that scenario, 12 to 18 months is a very, very, very short timeline. I think from a practical standpoint, can we make and test that vaccine in a quicker time? I absolutely think we can. I mean, there are innovative ways, and ideally without compromising any risk. But it's going to require some, forward thinking by the regulators and some pushing by companies in the space. I hope that answers your question.
Yes, thank you.
Thank you. Yeah, we have a lot more questions piling up here, both in terms of hands raised and in terms of chat questions. I'm going to take Wilson, and then I'm going to take Ravi, and then we go chat hand raise again. So Wilson, you go.
Hey, John. Yeah, I just had a question about why the vaccine is so well tolerated compared to the other vaccines. What are is it because of the metabolites? What's the basic reason?
Yeah, it's actually relatively simple. So DNA and RNA delivery platforms typically depend upon being taken up by the cell actively by endocytosis. And then they use these positively charged lipids to sort of stick to the membranes and break them open. So it's that biophysical breaking open or disruption of the membrane that causes the toxicity. And at a very low dose, it's fine. But as you increase the dose, and you can't really control the dose when you're injecting something or even systemically, the issue is that in the place of the highest dose, that's the toxicity that matters the most. So you get these dose-limiting toxicities that just makes it impossible to dose to higher levels. And every technology is based on this original technology that Moderna and everybody's using, and it works really well for siRNA, for instance, for certain things, but does a very poor job of delivering DNA.
Thank you.
All right, Ravi, you're next.
Great, thanks. Couple quick questions. One is sort of, does it look like it'd be possible for you to do a combined phase 1/2 trial? I've seen a couple of companies that are trying to do that just to try to accelerate time to market. Is that something to be feasible in this?
Yeah, so we're looking. Yeah, so we're looking at every opportunity. I think we've already had conversations with the regulators, and we'll have the phase 1, phase 2 mapped out. Whether they're a combined trial or not, there's some risk to doing a combined trial as well if you don't hit your certain efficacy endpoints in the phase 1. But certainly, I think everybody's motivated to move quickly as soon as we know from the phase 1 portion of a trial that it's safe to move to that phase 2 portion. So I know there's going to be some flexibility on the regulator side.
Mm-hmm. Great, that'd be great. And then when it wasn't entirely clear sort of what DNA fragments you were delivering. It looked like you were going to use the specific targeted epitopes from Epivax. Is that right?
Yeah, so we have a number of different things we're looking at. So we're taking we've started with the approach that others have taken, expressing the full spike protein, although we're using engineered versions of that based on Epivax's input. And we're also looking at optimized just fragments of the spike protein. And again, we want the vaccine to do two things. We want the vaccine to generate neutralizing antibodies that will prevent viral uptake and give this sustained protection, but also a cell-based response. So we want an active T-cell based response, and these are smaller epitopes that Epivax has designed. We've got a few of our own that we've come up with, and so like I said, we got about 24 candidates that sort of mix and match different aspects, and we're going to be sort of pitting them all against each other and picking a lead shortly.
So, and are you able to then combine a lot of these different peptides within a single vaccine?
Correct. We have one version that has, I think, about 32 epitopes in a single vaccine. Wow.
Okay. And are there limits in size? Like you're looking at neutralizing antibodies, you don't encode the antibody into the DNA. You encode something that then provokes the antibody response. Is that correct?
That's right. And one of the challenges is sometimes you get a lot, if you give the whole protein, sometimes there's what's called an immunodominant region that will produce a lot of antibodies, but actually won't inhibit a viral uptake. So that's why we have a lot of different shots on goal, because we want to make sure we maximize the production of these neutralizing antibodies.
Mm-hmm. Great, thank you.
All right, let's see how many questions we can still fit in, because there's more and more hands popping up. So next one up, we had Mira Flor. Mira Flor, do you want to ask your question? Okay, your mic doesn't work. Okay, then I'm going to ask you a question. How adaptable is this technology to future potential viruses?
Yeah, that's a great question. So we're thinking about emergent coronaviruses, because SARS, MERS, and now SARS-CoV-2 were all coronaviruses. That's why we think it's really important to build in this pan-coronavirus protection. And so we're incorporating key aspects of the nucleic acid protein into our candidates. And so ideally, if we can produce a pan-coronavirus vaccine, and there are models to test MERS and SARS as well, we should know that pretty quickly. I think that would be ideal. We also have plans to basically take this approach and apply it to other infectious diseases. So obviously, seasonal influenza is a big one. Thinking about HIV, thinking about many of the other dangerous viruses, Zika, et cetera, that are circulating in the world. Absolutely.
All right, great. Then next one we have Steve, please.
Hi. Can you hear me?
I can.
Okay, good. So my question is a little bit off from the other questions, but there have been reports that the virus originated from a bio lab in Wuhan, or some kind of lab. And so the question is, some people say it was a natural occurring virus from bats, and some say it was actually manufactured as a weapon. So I was wondering your opinion on that, and if it was manufactured for a weapon, would this vaccine work?
Yeah, I'm not going to weigh into that too deeply. I would say it's a pretty crappy weapon. So with a less than 1% kill rate, the issue is it's really easily spread. So we have quite a bit of data now on how fast it mutates. It definitely mutates, but it mutates a lot slower than some of the other viruses we've seen. And I think by taking our approach of using a lot of different epitopes from different parts of the coronavirus, I think we should be able to accommodate any mutations that happen, because again, there'll be multiple epitopes that the antibodies will be produced against.
Thank you.
Sweet, thanks. Creon.
Yeah, I concede my place to someone who hasn't talked, if you want me to, no problem.
Okay, then Josh and then Creon.
Okay.
Hi, can you hear me?
Again.
There's a substance called myrcene, and it kills tumor cells. And I want to know if you're familiar with that. And
So what is it called again?
it's called myrcene.
Myrcene.
Yeah, it's a substance from a quahog clamshell.
The longest lived creature.
Yeah.
Actually, I'm not familiar with it. Tell me about it.
My dad first started in Time magazine in 2002. I looked into it. It was done by a researcher in the 60s and 70s. And I think she was trying to synthesize it because it takes a lot of clams. But I've been trying to find a cancer group who could look more into it for quite a while.
You're welcome to send me an email.
Okay.
Hey, I've got two. One is the programmatic one, and one is a basic technical one. So the programmatic one is, have you briefed NIH or CDC or anyone high up on this? It sounds very promising. Presumably they're soliciting input. Have they gotten your input, and have you gotten a response or a reaction or anything?
Yeah, so we're actively engaged with both many of the different American government agencies. Also, because of our Canadian connections, I'll just mention that because Trump blocked the sale from 3M of N95 masks to Canada, the Canadian government is actually pretty freaked out that they won't get vaccines. So we may have an opportunity to get Canadian government support as well for our manufacturing. So yeah, absolutely. So we're looking at all avenues for support, and we're also engaged with both Health Canada and the FDA as far as planning out our clinical development pathway.
Okay, and then the technical question is this. I mean, excuse my ignorance, but how does the DNA vaccine actually work? And what I mean by that is, does it somehow get itself into the nucleus and then get transcribed and then make these proteins, and then they get presented, and then they get a classic adaptive immune response, or is it some other thing that's going on?
No, you're absolutely right. So for DNA to vaccine to work, our Fusogenix platform will fuse directly with the outside plasma membrane of cells, put the DNA into the cytoplasm. The DNA plasmids we're working with have nuclear localization sequences in them, and so we get very effective shuttling to the nucleus, and then they use the machinery of the cell to make the proteins that we want.
Thank you.
Believe it or not, we have time for one more question. So if anyone still has one in petto, you may speak up now.
I have one.
Okay, I have one. Oh yeah, you go. Go for it.
So is it accurate to say that the Fusogenix platform was originally developed for senescent cells and you've redeployed it for vaccines? Is that right?
So we originally developed in my lab in Canada, and so we're working with about 18 other companies on their various assets moving forward. I would say one of the most exciting applications is definitely with Ocean and longevity.
Yeah,
cool.
I agree,
Thanks.
and I shared the talk early up in this chat on that, because John was at our previous longevity investment salon. Jim, you were at that one too, I think.
Yes.
So you may have seen the talk as well.
Yeah, I had a 2-minute pitch, was significantly shorter.
Yeah. but short and sweet. All right, I have one last question. So we had the other day we had David Mannheim here on trying to accelerate, with a proposal to accelerate vaccine production. So do you think, you showed a slide on the manufacturing process and stuff, but what's the timeline there? Can we prep somehow on that end, and if so, what needs to be done there, because that does seem to be a bottleneck that, for example, I wasn't aware of until he gave the talk.
Yeah, no, I mean, and Bill Gates sort of summed it up right as well. We need to get building the manufacturing capacity now to be ready for 6 months, a year, 18 months down the road when we're ready to make, potentially billions of vaccines. So fortunately, the microfluidic platform that we're using is extremely quick. So we can make 25 liters in less than half an hour. And so, and it's conceivable that we can start, we can do it under continuous manufacturing. We can do it in parallel manufacturing. So I think it's pretty straightforward for us to get up to the amount of doses. A key inflection point for us will be that key readout for the animal data, showing that with a single dose, we produce the required immune response for protection and potentially active therapy of COVID-19. And with that, we'll be full speed ahead to produce that commercial manufacturing.
Hey,
And when can we wait for it? Or like, what's the date?
So the animal data we'll be getting in less than a week. Might take us a day to crunch it, but yeah, we're about 2 weeks away from having a really good idea whether the first candidates we've tested are going to make it.
Amazing. Okay, that sounds great. Well, maybe we can have you back. That would be lovely. Kriyan, did you want to say one last word?
One last question, since we have an expert on the line here. This is not about the vaccine, but I wondered if you had any ability to weigh in on this sort of, I call it fear porn, which is what I hope it is, that that there's all this reinfection and that adaptive immune response is ineffective against this virus. What do you think about that? How likely is that?
I haven't seen any credible information yet that reinfection can happen. I mean, we have seen it with some other viruses where you don't mount a really strong immune response. But I think based on the fact that we've seen a number of studies now that have isolated some really potent neutralizing antibodies, I think there's, this is probably more anecdotal than a real fear.
Thank you.
Oh man, this was fantastic. Thank you so much for being so spontaneously available and then totally acing this talk. I'm terribly excited for, I think, what's next down the line for you. I am glad that you're showing your email. If anyone is trying to reach John but can't get a hold of him and may kind of like want to contribute to the new round that they're raising, please reach out to me. And I just shared how you can join the next salons. Tomorrow I'm going to be doing one that's more like a brainstorm, so it's going to be quite informal. I'm hoping to get a lot of your input on the Foresight Hive Mind. So that's the kind of like more workshop type and more kind of like, focus group kind of like work that we're going to start on Saturday. And yeah, I basically want your opinions on what I'm missing, who else should be there, and kind of like what would be very specific actionable projects that we can tackle for ways in which we can kind of like create beneficial long-term futures from the current crisis. So tomorrow I really want to hear from you. For now, I thank you so much, John. I'm hoping that you'll share updates with me, and if it's viable and if it's useful, then that we have you on here rather sooner than later, hopefully. I wish you the best of luck with the data that's coming in. And yeah, we're rooting for it. And thank you so much. I hope you all have a lovely Saturday night. And I'll be, I think I'm going to have the video up tomorrow morning. So it should be on our playlist that I shared the link to in the chat room here. Okay?
Thanks, Allison. It was a real pleasure to be on, and thanks for accommodating us at such short notice.
It was awesome. Thank you so much. Bye-bye, everyone.